Experiments have proven that the coronavirus is now 10 times more contagious than it was when it first appeared in China late last year. Scientists attributed the change to the virus's ability to combine surface proteins into host cells. The international journal Nature said on the 1st, local time, that a series of mutations in the coronavirus that have spread around the world have been confirmed to be much more infectious in experiments on human cells and hamsters. In February this year, scientists discovered mutations in the spike protein that the SARS-CoV-2 virus binds to human cells. The mutant, named D614G, has since spread to Europe, North America and Asia, and is now found in most of the patients infected with corona. 1. Mutations increase virus infection. Professor Ralph Barrick of the University of North Carolina and Professor Pei-Yong of the University of Texas, respectively, have recently written on BioArchive that a mutant virus found in patients with corona has been created in the laboratory, making it much more infectious to human cells and hamsters, the journal said. Researchers at the University of North Carolina announced the results of an experiment in BioArchive on June 29 comparing the infectious power of the D614G with the normal coronavirus. The mutant virus did not differ in appearance from the electron microscope. However, more people have been infected with nasal and airway epithelial cells than regular viruses. In particular, the D614G mutant virus and the regular coronavirus were injected into epithelial cells at the same time, and 10 times more mutant viruses were later found in cultured cells. This means that the infection is strong. 2. Less defective and more productive during replication. Researchers at the University of Texas School of Medicine also told BioArchive on the 2nd of last month that the D614G mutations were more than 10 times more contagious than regular coronavirus. The researchers found 2.4 times more mutant viruses after 48 hours of lung epithelial infection tests than normal coronavirus. On the other hand, the virus's genetic material, RNA, was found similar. Considering the ratio of virus particles to RNA, it means that the D614G mutation has more than 10 times the infection power, the researchers said. Jung-Yang Sok, a professor at Kyunghee University, said, "The RNA, which is a component, is similar, but the fact that there are many virus particles, which is the final product, means that mutations are less defective and more productive during the cloning process," adding, "It is not just because of the productivity increase, but also because of the development of technology that doubles the ability to penetrate cells." 3. One-letter difference in the genetic blueprint. Protein is a form in which 20 pieces of amino acid are connected. Each amino acid is determined in the order in which 3 bases, the substances that make up the gene, are connected. In other words, life is made of the desired protein by connecting certain amino acids according to the sequence information of the gene. David Montpiori of Duke University in the US and Dr. Bette Korber of the Los Alamos National Laboratory confirmed in March that aspartic acid D, the 614th of the amino acids that make up the spike protein, was changed to glycine. The researchers named the mutant virus D614G as the 614th amino acid D changed to G. The researchers said the mutation occurred as only one of the 29,903 bases that make up the genes of the coronavirus changed. In other words, the final product is completely different from one letter in the design changes. The mutant virus was minimal early this year and began to spread to Europe, North America, and Asia in the spring. Starting in March, it began to appear in half of corona's patients and surged to 75% by June. In August, Malaysian health officials said they found a D614G coronavirus strain that was 10 times stronger and could easily be spread by super radio operators. 4. Increased binding force due to changes in spike protein. Specific causes of the virus' increased infection have also been revealed one after another. In a paper published in June, researchers at the Massachusetts Institute of Medicine said the D614G mutant virus has become better bound to host cells as the structure of the spike protein has changed from a closed form to an open one. Researchers at the University of Texas School said in a BioArchive paper on March 2 that the original form of the coronavirus was 20 to 30% less efficient than the D614G mutant in human cell infection tests. The coronavirus cuts the spike protein, combining one side with the host cell, and the other with the host cell's outer skin and the cell membrane of the host cell. A high protein cutting efficiency means that the virus's cellular penetration is stronger.